This lecture is largely about germination, but also about hormones that direct germination and growth. And we said before that plants do not have determinate growth. That means that you grow to a certain size, and that's how big you're going to be for your whole adult life. So I'm going to be 5.5 forever. And apparently, though, you start shrinking when you get older, so I might get shorter. Not likely to get taller unless one thing that can make you taller is apparently Pilates, because it makes your posture so good. But that's kind of an aside, because mostly animals have determinate growth. I mean, there are cases where there are fishes, for example, aquarium fishes that will grow larger if the aquarium is larger. If you have a small aquarium, they won't grow as large. But they won't grow for their entire lives. Rather, they will stop growing at some point. So plants have the capability of growing their entire lives. And growth is as a result of two functions. One is cell division, in other words, making new cells, and the other is cell elongation, which we saw with plants growing toward the light, and the cells on the back of the stem get longer, which pushes the stem in the direction of light. So yeah, can you germinate 10,000-year-old seeds? Yes. This plant grew from a 10,000-year-old seed. My goodness. Seeds can stay dormant for a very long time. These are the plant hormones responsible for such things as germination and growth. So these plant hormones are small molecules. They're produced in one part of the plant, and they move to another part of the plant and produce a response. They work in very small concentrations. These are the hormones of interest. Auxins. They're found in apical meristem. They're found in the embryos of seeds, and they're found in young leaves, and they generally move down the plant. We'll talk about cytokinins, Jibarylans, abscisic acid, and ethylene. Let's take a look at auxin. Lots of effects. Auxin affects phototropism, affects gravitropism. It has apical dominance. In other words, it will cut off axillary buds growing to the side. Lateral growth. It's functional in root formation and the development of fruit, and it orients the plant in an upright position. Auxins. Cytokinins promote cell division. Cytokinins are found in roots, and they move up to shoots through the xylem. They promote shoot growth. So auxins, lots of auxins, are opposed to the development of axillary buds. Those are the buds that are here that will grow laterally. They transport downward, and they favor rooting. Cytokinins, on the other hand, are opposed to root development. Lots of cytokinins, they will move upward and favor the development of axillary buds. So they have opposing effects. And the response depends on the relative concentrations of those two hormones. If you have more roots, you have more cytokinins. If you have more shoots, you have more auxins. And this is the effect. You start with a piece of plant tissue and apply a 1-to-1 ratio of auxin to cytokinin. A callus grows. A callus is just a bundle of cells. You apply a 10-to-1 ratio of auxin to cytokinin, and roots will form. High auxin concentration. If you have a high cytokinin concentration relative to auxin, so less than that ratio, shoots will form. So both hormones are required for root formation and shoot formation. You can use auxins and cytokinins to culture tissue and clone plants. Cloning is when you produce plants that are genetically identical to the parent plant. And this is done quite a lot in agriculture because some plants are desirable for their traits. Some plants clone themselves in nature, like ginger, for example, will just simply clone itself and send up a new plant from its stems. When you take a cutting of a plant, you might have done that at home. You can cut a branch off of a plant, put it in water, and some of those cells may become root cells and grow roots. So a lot of plants that we produce are genetically modified plants, genetically engineered. That's a different process. The genes are actually changed, or rather, they are transported or transplanted, rather, that's the word I wanted, transplanted to a different cell, a different plant. Jibarylans, also known as GA3, you'll find them in young shoots and in seeds. They move through the plant using the xylem and phloem roots. And Jibarylans promote stem elongation. Jibarylans will initiate seed germination and are involved in flowering. A dwarf plant can't produce Jibarylans. So they do not have the gene to do that. They're a mutant. A Jibarylan to turn seed germination on, but abscisic acid, or ABA, turns off seed germination. So Jibarylans are the go hormone in this case. So Jibarylans, interestingly, were discovered in a fungus. So there's a disease called the foolish seedling disease. It's a disease of rice, and it's caused by a fungus. The fungus is called Gibberella, and it produces Jibarylans, a plant hormone. What happens, and the reason that the rice is affected by this, is that the stems elongate quickly from the Jibarylans, from the fungus. They become weak and they fall over. And so the fungus then absorbs the nutrients from the dying plants. Since that time, we've found Jibarylans in plants. So Jibarylans promote cell elongation and rates of cell division. Here's a dwarf pea plant here, cannot produce Jibarylans, so it simply doesn't grow. Its stems are very short. But if you add Jibarylans to a dwarf plant, it will grow. Our responses to hormones depend on dose. You might be thinking about this for your experiments. How much is added? What is the concentration? What is the concentration? So this y-axis indicates the height of plants. These are dwarf peas that don't make their own Jibarylans. And this x-axis shows Jibarylan concentration. If there's no Jibarylans added, this is how high the plants grow. Add a little bit, they'll grow up to here. If you add more, they'll grow more. 1 and 10, they grow a little bit more, but they're starting to level off because there is a limit to how much Jibarylan you can add. Getting carried away. So an experiment that you can do, because we have Jibarylans that you can spray, is that you can spray the Jibarylan and measure the growth response. So that's an example of an experiment. You may be doing an experiment on germinations. How do seeds know when to germinate? What's the advantage? Well, some seeds need a cold period before germinating to ensure that they're germinating in spring and not in the fall. Some seeds need a dry period before germinating. And desert seeds need a lot of rain to germinate. What triggers it? What are the plant hormones involved? Here is a seed, a simple seed, a bean seed, a dicot. Dicot stands for 2 cotyledons. This is the cotyledon in the plant. Here. There's 2 of them. And this is the nutrition that will be provided to a growing embryo. This is the embryo here. The cotyledons are just starch, essentially. That's a dicot. And a monocot, which has 1 cotyledon. Here's the cotyledon here with the starch and the embryo. So in this case, in this case, I have to erase this because the nutrition that was in the cotyledon got transferred to the endosperm. Endosperm is just a nutritious mass of starch that's within the seed. So the seed may derive its nutrition from the cotyledons, or it may derive its nutrition from the endosperm. This is barley. It gets a nutrition from the endosperm. In both cases, however, it's starch. So seeds germinate, and when they do so, they're provided some protection by the way in which they emerge from the soil. So in this case, this is the seed coat, and it remains intact until the plant has emerged, and that protects the leaves. So starch that was within the seed is digested by the embryo and used for growth. There is no photosynthesis in the early growth of the embryo. What turns the seed on? What is going to make that seed germinate? Well, water. Water is of utmost importance. Jibarylans. Jibarylans will trigger seed germination. Amylase is really important. Amylase has to digest the starch to make the sugars available to the embryo. So starch is required, and it's digested into sugar. Why does the seed need to digest the starch? Well, it can't make its own food through photosynthesis. It just simply does not do that in the beginning of its life. So what happens when a seed germinates? Water's taken in, the seed swells up. The seed has to swell up. It has to break the seed coat in order for the embryo to emerge. The Jibarylans are produced by the embryo. In other words, the embryo is saying, I'm hungry. The Jibarylan moves to the aileron layer, that's the outer layer, and the aileron layer produces amylase, which is okay, yeah, the food is coming. Amylase moves to the starch, digests it into sugar. Here's the food for the embryo, and the embryo uses the sugar. Yum, yum. And then the embryo can start to make leaves with growth. Then photosynthesis happens, and it can start to make its own food. So another tool in biology is called the bioassay. The bioassay is a quantitative evaluation of the concentration of a substance by assessing its effect on tissues and cells, and animals and plants. So, for example, can we use a bioassay to figure out how much caffeine is in a particular soft drink? Okay, well, say I could quantitatively measure movement. So if I've had just a little bit of caffeine, I might be like, doo, doo, doo. If I've had quite a lot of caffeine, maybe 2 or 3 cups of coffee, I'd be like moving a fair bit more. And then if I had like lots of caffeine, I'd be moving like crazy. And if you can measure my movement, you can measure the amount of caffeine that I've ingested. So how much Jibarylan a seed produces can also be measured by bioassay. Here's barley on a starch media. No Jibarylans are added. It's cut in half. The endosperm half is placed in one petri dish. The embryo half is placed in another petri dish. In which plate will the starch be digested? Not here. There's no embryo because the embryo is the one that produces the Jibarylan. So how much starch will be digested? You can measure that. So in this case, we've dyed the petri dish with iodine, which has made it black. In this case, this is all starch. None of it is digested. There are no Jibarylans present. But in the embryo half, which produces Jibarylans, starch is digested. And you can see that here, where the starch is no longer present. That's a bioassay. And you can correlate the area, this area, with the amount of Jibarylans present. Another plant hormone of interest is abscisic acid, also known as ABA. ABA, and it turns off seed germination. So Jibarylans turn seed germination on, but ABA turns seed germination off. They compete, interestingly, for the same binding sites on DNA. And the response of the seed, again, depends on the proportion of the Jibarylans to the abscisic acid. Yeah, so in the desert, when there's a lot of rain, the ABA, the one that's inhibiting germination, is washed out of the seed and it germinates. So that's how the seed knows to germinate. Yeah. It also promotes leaf drop. So when trees or other plants drop their leaves, In the fall, that's a signal from abscisic acid. And it has another interesting function, which is to close the stomata of a plant. Stomata, if you recall, are the holes on the bottom of the leaves that allow water evaporation and also the intake of carbon dioxide. So if ABA is in the roots in large concentration, it signals that there may be a drought. So it moves from the roots to the shoots. It causes abscission so that the leaves won't be requiring water. And ethylene is a whole other signaling mechanism. It's actually a gas. And it's usually a growth inhibitor, but it helps to ripen fruit. It also assists with leaf abscission and fruit drop and the initiation of flowering. So it has many different functions. And thus endeth the lecture. We'll have a little discussion.